In this web seminar from mTuitive and the College of American Pathologists, you'll hear from a panel of presenters ready to share their experience and insights. Dr. Richard Eisen is a practicing pathologist in the Banner Health System in Phoenix, Arizona, and chair for the IHC Committee for Banner Laboratory Sciences of Arizona. Dr. Eisen has served as a member of the IHC Resource Committee of the CAP and as an advisor to the CAP Center Project on IHC validation guidelines. He was also a member of the CAP Center Project for Uniform Labeling Standards for Anatomic Pathology. Dr. Daniela Mihova is Director of Clinical Laboratories, Vice Chief of Medical Staff, and a member of the Board of Directors at Frederick Health Hospital in Frederick, Maryland. She's a member of the Frederick Health Cancer Committee and Precision Medicine Genomics Steering Committee. Dr. Mihova has spearheaded the implementation of Synoptic Reporting as part of the Commission on Cancer and CAP accreditation and worked on streamlining an electronic process for patients' genetic and molecular testing, along with clinical trial identification and enrollment. Dr. Mark Tuttle is Division Head of Pathology Informatics at Henry Ford Health in Detroit. He's a nationally recognized expert on pathology informatics and the use of synoptic reporting. His expertise spans digital pathology implementation, internet applications for laboratory services, laboratory information systems, business analytics, electronic health records, and informatics training and education. Eric Daly is Senior Clinical Product Manager for the CAP. He leads development of clinical content for the CAP electronic cancer protocols and provides expert guidance to vendors for integration into AP and LIS systems. With experience as a pathologist assistant and laboratory leader, he collaborates with field experts to create and refine data entry elements from the CAP cancer protocols. And last but certainly not least, Peter O'Toole has been a pivotal figure in the healthcare technology sector for over 2 decades. As president of mTuitive, he leads software architecture, spearheading product development of innovative healthcare software, including mTuitive Pathology, mTuitive Surgery, and mTuitive Insight. Mr. O'Toole's expertise in developing and integrating advanced software solutions with EHR and LIS applications has profoundly impacted the way cancer data is captured and used to improve patient care. I'm going to turn off my camera and turn things over to Eric Daly to get us into our topic. Eric, it's all yours.
Thanks, Ken. Well, as Ken alluded to, today we're going to be doing a multi-phase panel. We're going to start off our session today with how structured data is improving the diagnostic accuracy. We're going to look at what are the CAP cancer protocols, what are the electronic cancer protocols, and what is structured data, how it can be used and be beneficial in your use cases every day, and then look at some quality improvements in this first section. So when first looking at what are the cancer protocols are a form-based system that looks at clinical diagnostics in cancer. There's 101 of these protocols posted on our CAP website, specifically on the Cancer Protocol Templates website. They're built with other standards associations such as the AJCC, WHO, and ICD-03. When looking at how these protocols are created, there is multiple collaborations that occur to create these. The first collaborative partner is our Cancer Committee. These are our expert authors who author the content that's present in the cancer protocols. They serve as liaisons with AJCC, WHO, and ASCO, just to name a few of the other organizations we work with for the content. We then have our Pathology Electronic Reporting Committee. This committee is designed to look at the structure and the modeling that goes behind the scenes. They also help serve as resources with our vendors in the implementation of the electronic cancer protocols. And then we also have the Cancer Protocol and Data Standards team. This is the internal CAP staff that works on these cancer protocols and the electronic versions of them. There's many other councils and committees that also help author these. Just want to make sure we're representative to all of them. When looking at the electronic cancer protocols are an electronic format of the cancer protocols. They use a XML structure known as structured data capture. That is a unique software that we have co-developed with many other partners, such as the CDC. When you're looking at this screen right now, on the left-hand side, you are seeing our reference implementation. This is actually looking at the most current breast resection protocol. This serves as kind of a tool to help our vendors when they implement. On the right-hand side, you're seeing mTuitive software for this and showing how it would look in an electronic format in your APLAS. So when looking at the ECP, it is a license-based software, and it also is designed that once you purchase the license-based software, that it gets integrated into your APLES workflow. Some of the benefits that occur with the ECP is that it'll help you with your cancer reporting. It serves as a helper for accreditation compliance, allows you to have structured data to pull from and pull queries to look at downstream analytics. And then it also assists you with any further use with cancer registrars and other downstream users that may want to see the diagnostic material. When looking at implementing the ECPs at the CAP, we kind of look at this as an ability to collaborate with not only our end users but our vendors. Such relationships like we have with mTuitive allows for the perfect harmonized implementation, as you're seeing right now on the screen, showing how their form is interacting with how our content is designed and the metadata behind it to allow for an efficient form filling ability. So with that being said, I'm going to actually turn this over and have a question for Dr. Mihova. How has the adoption of structured data improved diagnostic accuracy in cancer care, and what tangible benefits have you seen in your practice, Dr. Mihova?
Thank you, Eric, for the question. So we adopted electronic cancer reporting 2 years ago with mTuitive, and it's still a standalone program, which is still not implemented in our LIS program, but it's on its way. And immediately what we identify by adopting the electronic cancer reporting is that it's very efficient, easy to work, intuitive, and also keep us compliant with the CAP checklist. It prevents omitting important required data from the cancer protocols. Also, it helps us to be always up to date with the tumor staging and the newest classification. It helps us with the CAP accreditation. So far, it's 100% compliance, and also with Commission on Cancer accreditation. When they come for inspection, there are a certain number of random pathology reports which are checked for completeness, accuracy, staging, and so far, again, we have been 100% compliant. The tangible benefits, since the program is so easy to use and so easy, such easy flow with completing all the sections, we have noticed that there is a faster turnaround time and improvement in the pathologist sign-out turnaround time. Also, we have received compliments for easiness and clarity of our reports from physicians and from the tumor registry. It's easy for them to identify the data which is reportable and to match patients to clinical trials or for treatment. Additionally, I find that the auto staging, which is with the program, is helping for patient safety because it prevents typographical errors in the staging of the tumors. And in this way, it keeps us with making sure that the patient receives final correct diagnosis and treatment. And because of the automatic updates on a regular basis of all the cancer summaries, we have noticed that we have more time to focus on diagnosis, on clinical work. Before, we always had a pathologist that goes and checks on a regular basis the cancer protocols, make sure that the templates are accurate, that everybody is notified. Right now, everybody is on the same page, and it's provided automatically on a timely basis. Additionally, we have had more time for our QI department and our histology team, again, to focus more on clinical and patient care rather than reading reports and making sure that all the required elements are included. So far, the experience has been great. And thank you very much for the question.
Well, thank you, Dr. Mihova. So moving on to the next panelist, Dr. Tuttle. How has the adoption of the structured data improved diagnostic accuracy in cancer care, and what were the tangible benefits for your practice?
Well, I would say the simple answer, Eric, is that it's transformed our clinical reporting. Probably one of the most common reasons for a report amendment after a good diagnosis was that we missed something in the staging. We either omitted a feature or we had a slight typographical error, as Dr. Daniela was referring to. And so these errors of omission and commission are really pretty difficult, especially if you look at reports like the breast cancer checklist, which has got tens of elements. If you look at the electronic version or you look at the paper version of that, it's almost 40 pages long. It takes up about 2.5 pages of the pathology report. So for a pathologist to get that right with pen and paper is, I would say, almost impossible at this point in time. And since you aren't omitting things and you're not entering data incorrectly, the opportunity to do auto staging was absolutely critical to deployment. Now, that's an assumed piece of mTuitive right now. When we started using mTuitive 20 years ago, we did not have that. We still had to build those things out. And when we first started to build those calculations, it took a tremendous amount of time and effort. We entered into our partnership with mTuitive with the CAP e-checklists probably 5-6 years ago. Prior to that, we were building them all and maintaining them all ourselves, and it has been a tremendous value. It has decreased so much time and effort on the part of our informatics team, on the part of our pathologists. We still have to validate them whenever we bring a new checklist in. Someone has to go through it, but it's a way more efficient process. Just the data entry efficiency alone is worth the price of admission, in my opinion. It just makes it so much easier for people to put this data in. And of course, the fringe benefits are that you're actually getting true data out of the system. So when you put in a number, it's a number. And so you can do things like calculations off of that. You can do averages. You can do counts. And we'll talk about that a little bit later in the webinar. But as I've said many, many times, it's been transformative. And I always allude to the fact that our pathologists say that this is the best thing that we've done with our information technology other than not having to use the typewriter. So really, really well accepted and high impact on our patients.
Thanks, Dr. Tuttle. Mr. O'Toole, when reviewing quality metrics and performance indicators, how can structured data help, and how does mTuitive support that?
Sure. If we could go, Ken, could we bring up slide number 10? And yeah, Eric, Synoptic Reporting on its own, when it started out on paper and eventually went to sort of electronic paper in the form of Word documents or word processors, I think was helpful in, making sure that reports were complete and that there was some standardization to pathology reports and that important data elements were included in pathology reports. But as Dr. Tuttle just alluded to, once you have that process down, once we move to more real structured data reporting, that enables data as a byproduct of the pathologist doing their normal report. And so, once you have highly structured data based on a standardized source like the CAP's protocols, then you can do a lot of really interesting things. Instead of having to manually abstract data when you want to do a study or check for quality or track performance, you can use standard reporting tools and operate on the data in a database-like fashion. So this is an example from one of our customers, the province of British Columbia, one of the probably largest implementations of synoptic reporting in the world, where the whole province has been using CAP standardized data through mTuitive for years, and all their different health authorities and labs throughout the province store their data in a central database. And a very innovative customer, they were able to create their own dashboards that track, I would say, 100 different quality metrics. And those metrics aren't limited just to helping out, tracking quality in the lab. They can also be used as performance indicators for quality of care. There's surgeon scorecards, and they can compare across sites and end users. So, that's one example. Another is, there are many types of reports you might want to build that might help with, matching patients, for specific treatments or trials. There's a lot of possibilities, and just featured in the background here is mTuitive Insight, which is a new user interface mTuitive's created to do reporting on CAP-like data. And there's a bunch of reports we've identified there that are helpful to folks, and many of which are centered around CAP compliance and COC compliance, as well. Both the pathology and surgery requirements are much easier to comply with when you're tracking all that information as structured data. So again, we talked about data being a byproduct of using a structured synoptic reporting application. And when you do that, you can also automate treatment. You can automate, the processes that go into treatment. So for example, you can automatically take structured data and get it to the cancer registry. You can populate the oncologist's staging forms or other parts of the oncology workflow using the data collected in pathology. And again, you can do the opposite as well. Perhaps, pull data in from other sources, such as, reference laboratories, pull in next-gen sequencing results and things like that into your intelligent, CAP protocol type application like mTuitive, and it'll help you do additional, more intelligent things like staging calculations, identifying patients and pinpointing their specific, profile of disease. And then ultimately, all that data can be really helpful as well for research. So moving on to the next question for our panelists. For Dr. Eisen, how do you ensure that specific data points from pathology reports are available to your oncology colleagues for treatment planning? And do synoptic reports improve the way this information is passed?
Thank you for the question, Peter. Just to follow up on Dr. Tuttle's comments, in our journey in our health system, we went from, and I'll pull out the book, and paper, which was very laborious, and our pathologists either typing these things in themselves or dictating. And within that, the same problems we had where corrected reports were needed for omissions and errors. So having this, the mTuitive tool has improved efficiency dramatically, with structured reporting, structured data, and we've been able to accomplish that. One of the things we encountered in our information system was the fact that the synoptic report could be placed in different portions of the pathology report. So it wasn't initially, when you clicked it, it would go into one area initially. So in some cases, it was in the microfield. In some cases, people expanded the diagnostic field, and it was listed there, and it was huge in the report. So with standardization of that process and getting it into a field where we wanted it, which is our comment field, and then the end user, the oncologists, nurse, the coordinators for tumor boards, et cetera, know exactly where to see it in the report. So all of that has brought in efficiency and the fact that the data is then available to oncologist, surgeon, et cetera, in the right place, whether they pull up the report, in the medical record, or it's a printed report. And as you alluded to, the ability to extract reports, like if we want to extract just the synoptic report into one report, that could be used for a given tumor board where those patients that are listed, one can extract the data, not have to have the entire pathology report, to go through. So on a tumor board basis, that I think that helps the folks have the data when cases are discussed. Thank you.
Thanks so much. Do you find it cuts down on the back and forth between, let's say, an oncologist or surgeon getting results and then having to come back and inquire about, information that, that may have been left out?
Yeah, no, that's a very good point. Yeah, I think it has. I think the number of calls that I get regarding our reports have dropped dramatically since I joined the group here in the 9 years I've been here. Cool. Yeah, the calls we get were more about ancillary testing and where that resulted, but not the synoptic report.
Awesome. So, in Dr. Eisen's practice, there's they have Cerner for their EHR, Oracle Cerner, and in that case, the data is able to be pulled automatically into the oncologist's forms for staging, which is something we also do with a couple of other vendors, including Meditech. And in that case, there's a cooperative, willingness across, different vendors to make sure that the data you're collecting when you do a CAP protocol is going to be useful to other clinicians, downstream. And I just wanted to point out, I think that I remember the folks at Cerner at the time when you implemented this saying that they didn't understand why all of a sudden the usage by pathologists of the CAP protocols had gone up like 40% out of nowhere. And all of a sudden they were doing more biopsies and other like non-required protocols. And it turned out that it was because the oncologists had just got this feature, and the pathologists really, once they knew the oncologists would have access to this and it would help them, they started using synoptic reporting even more than they previously did. And that number might be anecdotal, but it was a big jump if I'm correct about that.
Absolutely. I think, it was a ramp up period, like getting used to any new technology or with the pathologists and getting everyone on board just jumped up the number of cases. And now it's essentially 100%. Just to tell you, I remember in my former practice when we first started using synoptic reporting before the E format, we were cited on an inspection for the cancer center because one of the pathologists refused to use synoptic reporting. And that was unfortunate, but it was then corrected quickly after that visit.
Gotcha. Excellent. Well, thanks a lot, Dr. Eisen. So another question we have for Dr. Mihova is if you could discuss the impact of structured data on staging and molecular profiling and precision medicine and how this is helping.
So as I said, currently we use the mTuitive separately. We copy and paste the report and we put it in the pathology report. Additionally, our hospitals initiated precision medicine genomics program. It launched 2 years ago, and we work with multiple reference labs to import discrete data into our LIS system. With this said, we still don't have the option to incorporate these results into our pathology reports. And why I think that's extremely important is you have completeness of the diagnosis, because you need the molecular and genetic reports and additional immunohistochemical reports, not only for the oncologist, but to make the diagnosis, to have the prognosis, to decide the treatment, or potentially put the patient in the right clinical trial. Additionally, to incorporate the entire data in the pathology report is very important for the patient itself. When they go to multiple other opinions, they carry the whole report with themselves instead of having fragmented pieces of information. And it's actually, I'm very thankful for inviting me to this webinar, because it also gives an opportunity to think about the future and how we can use all this information, which is extremely valuable, how we can incorporate it to to make our life easier, our patient treatment better, and help overall our population. So in the future, how I would like all this incorporation of tumor reporting and molecular genetic data to be able to easily derive retrospective reports, concurrent reports, and prospective. With this way, like with the retrospective reports, we can learn more about our patient population. We'll know what type of tumors, what molecular abnormalities we have in, let's say, in our area here in Frederick, in Maryland. Be able to assess our quality as physicians or communication with other disciplines, like, for example, radiology and pathology correlation. If we overutilize molecular data or underutilize, how is the correlation between, for example, let's say, finding the aspirates of the thyroids and the molecular data. So the opportunities a lot and will give us better understanding of what we have done and what we can improve, or we can keep the same. With the concurrent QA reports, we can identify patients which need current treatment, like EGFR mutation. This patient can be flagged and we can notify the clinicians and the oncologists on time. We can capture discrepancies, like if we have, for example, a low-grade breast cancer with ERPR negative results and HER2 positive, that will signal us to go and identify it and look more if these results are true and accurate. And the prospective reports could be identify patient for future medications, for clinical trials, and to overall see the progression of the disease and the diagnosis and treatment. And thank you very much for the discussion. It's really very helpful.
Thank you. A couple of thoughts based on what you said. That effort to get as much structured genomic data from different reference labs at once is, I know, something you've been working on with Meditech, which is awesome. I think a rising tide lifts all boats when it comes to interoperability across laboratories, and structured data. And I know we also work closely with them and are doing some similar efforts and hope to help each other in terms of being able to get discrete data from multiple different sources, into pathology reports, and then out from pathology reports back to the folks who ordered the test, the surgeons, the oncologists, and so forth. This slide I brought up toward the end of while you were speaking, just to show an example of, once you've collected this data, you mentioned, triple negative patients, we can come up with a list of those patients so that a research coordinator or someone in the cancer center can follow up and make sure that they're getting the appropriate treatment. And forgive me if I jump around just a little bit. Another one, the ERPR HER2 reports for quality purposes, again, using that biomarker information at in a database ready way to automate some of the reports you need to do for CAP. I think you may have touched on that real quick. And I wanted to go back up a little bit. This shows, finding clinical trials for patients and matching them based on some of that information. And this is in the Meditech system, which I think is a great use of that type of data, for precision medicine, and something you're probably familiar with. And I'll just back up one more slide here and just talk about, for just 1 minute, CAP and mTuitive and Banner Health, where Dr. Eisen is, and Caris, the laboratory, have collaborated on, a proof of concept that we're now going to be putting into production that involves automatically shipping results from Caris to Banner to pre-populate the CAP protocols through mTuitive, and to really take out all that redundant data entry and enable, additional, more precise ways of doing synoptic reporting. That'll obviously add to the types of calculations we can do, the types of queries and patient matching and things like that. And that's an effort that we really encourage the labs and the vendors and everybody to come together and make sure that can interoperate, basically no matter who you're using for your outside labs, your internal labs, and also for your reporting vendors and LISs. We want to make that all work in harmony. On that note, let me move on to our next panel question. For Dr. Tuttle, how does synoptic pathology data fit into the overall cancer research data strategy for you?
Well, it's a really good question and a great lead-in, Peter. I'm fascinated to see the auto-population coming from Caris going into mTuitive, because that's something we have dreamed of forever. This was really all enabled when mTuitive went to their newest platform, which, using web services and structured data, allows these structured data files to be readily externalized and then interoperable between systems. We started to use that web service when we got that version of mTuitive to ship our structured data as structured data to our enterprise data warehouse, which is the back end of our Epic EMR. We're Epic users. And we have now populated 22,000 synoptic reports into our Epic EDW. If you look at that, there's over 4,000 breast reports. So what does this give you? This gives you an incredibly rich database to look at outcomes. And as we move to things like value-based care, the value of pathology for the organization is demonstrated by these types of workflows and these types of workflow processes, where you can see how your patient population actually performed in various situations. Of course, and as alluded to by one of the other panelists, the correlation of the staging data in pathology with the cancer molecular data is a fascinating area for data mining. So very recently, we have been bringing in the data from our pipeline. We do most of our genomics testing in-house, but we use a third-party genomics pipeline to bring that data in, and it is coming in also as structured data, and that has also been being populated into our EDW. So it doesn't take a stretch to say, okay, let's look at patient outcome, let's look at the genomics, and let's look at the staging, and is there something lurking there that tells us what we could have done differently to get a better outcome? Why were the costs of care different? Why was patient outcome different? Why was higher stage better survivor than lower stage? I mean, there's all these variables that we really don't quite understand yet. And I think we'll get into this a little deeper when we go further in the conversation, because, of course, this is where things like AI can come in. So what you're seeing on the screen here is a very simple diagram where when we work in our CoPath system, the CoPath system immediately fires off a data message to the enterprise data warehouse that receives structured data into the warehouse that we can then use. And on the right-hand side, you're seeing one example of a pathology. I believe it's a prostate case. And you're seeing all of the different parameters of the structured staging that went on in mTuitive. And this is an Excel front end. So obviously, if it's an Excel front end, you can now start to ask questions across patients. And it's very easy for us to do things like, what was the average size of a T4 prostate cancer? Questions that were very difficult to answer out of the LIS because, of course, those weren't numbers. The other thing that we can do is we can populate other downstream systems. So pictured here is actually population of our biomaterial tracking system, in other words, our biobank. So we can dump this structured pathology staging information into our biobank to allow us to get better detailed information around tissues that researchers want to do work on. And I am very envious to see Dr. Eisen automatically sending this stuff out into his EMR, which is something that we've wanted to do for a long time. Epic has been a little bit of a roadblock. I can't say that they have been preventative because we have not pushed that hard on it. Frankly, a couple of other pieces had to happen. The big one being the Epic's genomics module, which we have just started to populate with structured genetic and molecular information. So what I see coming is a constellation, a universe of data related to cancer care and team communication that allows us to better understand the differences in outcome and how that impacts patients going into the future, and how we can improve our precision medicine programs, clinical trials. The road really never ends. It just keeps carrying on into the future. So I hope I addressed that well, Peter, and I stayed a little bit out of the AI stuff for the moment.
No, that's fantastic. And, AI is also a great thing for us to discuss, and maybe we might even get to that in the next section, when I think Eric's gonna take over and talk a little bit about the future. But that's really great to see what you did and to see how, you're able to integrate data from different sources, include it with Epic's data. It's really an awesome, an awesome case study, and what you can do with structured data. So thanks very much for speaking to that.
I also want to mention that before the webinar, I discussed a little bit in detail with also our precision medicine genomic program staff and the cancer registry. And they also want to be included in the information that is associated, molecular information and tumor staging, especially if we have early first diagnosis cancer, which has to be reported to the state. It's right now, everything is manual. It can be extracted and compiled as information to be sent to for reporting. And from precision medicine program, identification of all the patients with a stage 4 disease and if they qualify for next-gen sequencing also.
Okay. Excellent.
So our next section, our third phase of this panel, is really going to look at what's next. And I think Dr. Tuttle really explained it pretty well when he says this is like a universe of information. It really is. And we're moving to that next stage, which is what do we do with it next? So kind of we want to look at like how do we embrace AI in this? We want to look at how we can leverage digital pathology workflows, and then we're going to kind of follow up with what is coming next from the CAP with this as well, and our partners and vendors that we're working on with this. So I'm first going to ask a question to Dr. Tuthill. Dr. Tuthill, with the growing adoption of AI in healthcare, how important is concept standardization across different reporting templates, that universe as you were referencing before, and what challenges have you encountered in maintaining the consistency in the structured data?
Yeah, really good question. I think in the early days, this is what drove us because we realized that you could say things 5 different ways and that we couldn't compute on that. But as soon as we started to standardize text, and I'm not even talking about synoptic reporting yet, I'm talking about standardized text for things like microscopics or gross descriptions, et cetera. As soon as we started to do that, we now had computatable information that we could use as we go into the future. Obviously, e-checklists and entering e-checklists with tools like mTuitive take this to the nth degree because you are using consistent terms. I mean, not only does this help our surgeons understand what we're talking about or where to look in the report or what we mean by the report, but the computer can look at this and the computer can start to do very interesting things. So if you do not have common terminology, we're completely screwed in our ability to do intelligent data mining. Not to mention the opportunities that we have in AI for other aspects. So when we think about AI, there's really 2 things we need to think about. One is how do we use AI to mine data, find things that we might have missed, to combine things that we didn't as humans see relationships between, because the computer can look at a lot more things at once. But on the flip side, the computer can generate information, i.e. generative AI. This is our whole ChatGTP world we're living in right now, and can do that consistently. So if we have inconsistent data, we're going to get inconsistent generation of AI. We're going to get inconsistent combining of that information. Once we have consistent data entry, we now have the ability to use generative AI to take something like an mTuitive staging report and turn it into a pathology report that is done in context. And that even includes what people are doing right now, which is creating more understandable reports for patients. But if the content is all over the place, you can't reliably use those generative AI tools to do that work because they will make the usual mistakes that they make. One of the key problems with generative AI is it fabricates. If it doesn't have the answer, it just makes one up. And I have myriad of examples of that. And then, of course, that's really cool stuff, and generative AI is great. But where we're really going to get the bang for the buck, in my opinion, is when we start to apply AI across these knowledge domains to the data that we are getting out of our structured data reports, to the data that we're getting out of our structured oncology and molecular data, and to the data that we're seeing in our pathology images. And when we combine all 3 of those data sets, we're going to have novel discoveries that we don't even recognize yet. In fact, I think there's a fair amount of literature starting to emerge about things that the computer sees in histology that we don't see with our human eye that actually is very important to things like outcomes, patient care costs, so forth and so on. And it's only by combining these intersections that we can recognize what those opportunities are. So I'll pause right there, Peter. I hope I addressed what you were asking.
That was perfect. So moving next, I'd like to ask Dr. Eisen a question. And Dr. Eisen, I think kind of set you up perfectly for this, but how do you see digital pathology workflows interacting with artificial intelligence now moving forward?
Thank you, Eric. Yes, that's a very good lead-in, and thank you, Dr. Tuttle, for that. So on the screen, you can see this is just one of the tools we've been working with our AI partner in our digital system. So it's a breast cancer. If you look at the upper left, there's a low-power image of the cancer section, and in the lower right, you have, with the AI tool turned on, this is for mitosis identification and quantification. And in our initial experience in looking at this, it's quite accurate. So it will in sort of the reddish hue or purplish outline, it highlights a hotspot of mitosis for you. You can go right to that area, and then it actually puts each mitosis in a little box for you. So you can verify those. You probably see the three in the screen in the upper part. And then the analysis tool gives you a quantitative number. So it'll give you mitosis in the hotspot, and it'll give you an average mitosis of the whole section if you want that. But if you count, so it's very accurate, very quick for the pathologist. It improves efficiency for us when we use a tool like this. And ultimately, working with a partner like mTuitive is to basically take this data right from the AI tool in the report, populate it into your synoptic report. So as you put in, your grade, your mitosis count, you can do it very quickly rather than, the counting we do under the microscope or even on a digital slide manually, and then having to write that number down or remember it and put it in. So these are the kinds of tools that'll be very accurate and efficient. Quantitative measurements for just not only like tumor volumes, tumor measurements, margins, all of that data can be quickly entered into a synoptic report. The other thing we've begun to look into with the image management systems we're looking at to ultimately use across the system is the fact that some of these are now so robust where they can bring in data from anywhere in the medical record into the workspace. So I think that's been one of the ultimate goals of information technology is we have so much data from all these departments, different aspects of the medical record, radiology, et cetera. Can we bring it into one workspace where with one click you can get that? So bringing in radiology images, not only just pathology images, and then also data and reports into the workspace within the image management system will make pathologists make us tremendously more efficient. I think it'll improve the way we discuss and document management plans, diagnosis, staging, et cetera, management plans for conferences, tumor boards, and for teaching, et cetera, and training, training residents. All of that will just be tremendously improved by using AI tools like these. Thanks.
Great. Thank you, Dr. Eisen. That was an awesome answer. So to kind of end the panel discussion, the last objective here is just to kind of go over where we stand next at the CAP with the ECPs. We're looking at a few things. One of the first things we're looking at is how to incorporate AI potentially into our protocols. And we're working with the AI committee, the cancer committee, along with the pathology reporting committee into this more, trying to identify areas where we could possibly populate diagnostic information. When is it right? When is it wrong? Trying to, I think, first establish the rules of this, and then from there move into how we could work with vendors and partner with people like PedoTool and mTuitive on how to help support them in defining some of these AI algorithms and rules to use moving forward. Another project that we're working on very closely is our SNOMED project team. We're looking to develop a full concept map for all of our unique identifiers mapped to SNOMED concepts. That process is underway. We're hoping to have that first release of that this year. And really, the idea behind this is to really develop an ontology for helping people doing the data mining that I think several of the panelists have mentioned today. And then finally, the last exploration we're looking into is digital imaging and how can we annotate digital images like Dr. Eisen was speaking about with the CAP cancer protocols and moving forward with that process. So with that being said, I think we're ready to move to the next step. Ken, I think you can take it over if you'd like.
Fantastic. I will indeed. Our panelists have been kind enough to share their emails if you want to get in touch with them. We have a number of questions that have been submitted here. We'll get through what we can in the remainder of the hour. We'll take any remainders offline, get back to people as necessary. I'm going to start with one that was specifically addressed to Dr. Eisen. You're on the spot. It says, did the increase in synoptic reporting at implementation increase the number of billable procedures?
That's a good question. I mean, I do not deal with billing in my department. That's one of the things I'm glad I haven't taken on. or don't have that responsibility. I will say, I think for our coders, though, one of the things we struggle with, particularly even with immunohistochemistry results, where they were all over the map in reports, or they weren't specifically detailed enough so that bills were denied. So with synoptic reporting, which can include things like our MMR results, our breast biomarker templates, and structured, it's very easy for them to go to the report and verify the information and make sure the bill is correct.
All right. Thank you very much. I'm going to move on to, I'm guessing this would be best for Mr. Atul because it's specifically about mTuitive. And it's asking, how do you integrate the discrete data produced by mTuitive with Oracle Health?
Oh, great question. Yeah, so with Oracle, mTuitive has an integrated synoptic reporting solution that, once you have that in place, all of the CAP identifiers, or C keys as they call them, all the data is saved along with those C keys in the Oracle Cerner Millennium database, in addition to mTuitive's own system. And once that's done, the folks at Oracle have built essentially a smart app within their own oncology system that we showed a screenshot of earlier briefly. And that app knows how to, look at some mappings between the CAP C keys and their own internal identifiers in their oncology system and pull in the relevant data. And it gives you the oncologist user, the option to choose which synoptic reports on that patient they want to bring in and which data elements from those synoptic reports. So, for example, if there were 2 synoptic reports, they could take the grade from one and the T from another and the N from another, let's say. So yeah, it's an internal integration between those 2 vendors, and it works very well. It's a good byproduct of those kind of automated systems we talked about before.
All right, thank you. People are being really nice about saying who these should go to. So this one was addressed to Dr. Tuttle, and it's asking, would you agree that digital annotations and relevant AI clinical data should be implemented in the SSR underwater for better research and to avoid bias and data drift?
Yes. I think there's a lot underneath that, but I think the simple and short answer is absolutely yes, because if you're not structuring the data really, you're not going to be able to go further with any of this. So, this is like electricity to me. When we start to structure more and more of our pathology data this way, we will have better outcomes. So I've always dreamed of even implementing our synoptic reporting upstream into grossing and microscopic, so that it literally just flows into the synoptic reports, and that we're not typing in a gross and then going and figuring out what the values were for the synoptic report. So you can get it right once and it's right all the time. But every time you have to re-enter the data, you have the opportunities for mistakes. So yes, I think that's absolutely essential.
All right, here's a more open question, although I think it's probably for mTuitive primarily. Would you consider, or have you considered, setting up a way to flag critical or incoherent diagnoses? For example, indolent breast cancer, such as a tubular carcinoma with a HER2 positive result.
Any of the pathologists are welcome to jump in here. I can say that, the ability to create rules within mTuitive that would look at very specific values is something that, beyond what CAP may advise. Like, we'll have customers who want to look for a particular condition, let's say, some very specific, profile of a tumor that they want, like an alert to pop up, for example, and let them know, hey, when this happens, we want to order an additional test or whatever. And so that's one example we've seen, for sure, many times over the years, is working with a customer to implement like a very specific trigger, if you will, looking at that data. Another example, more retrospectively, would be like the list we showed earlier of triple negative breast cancer patients. So it may be an after the fact, we're looking for, not, like real time, but essentially we're looking for a report of sorts of all the patients that meet that criteria. In the first instance, it's interactive, while the pathologist is completing the synoptic report. So collecting this kind of data enables both use cases, and we've definitely worked with customers directly to implement those. And I think it would be great to see more of that, and even to encourage in some way, labs to share, what particular, types of alerts they implement and share them with each other would be great too, which we'd be happy to facilitate.
This is where non-generative AI comes to play, because you can start to use machine learning to help catch these type of contradictions in our own reports. When we're having to use rules and alerts, as Pete was just describing, that all has to get built, and that's problematic. But when we start to train these systems to recognize disparities on the fly, now we're at a whole new level. So I'll stop there.
True.
Okay, great. Thank you. We have a few more minutes, and I'm going to open this up to all of our panelists because this is a wide open question. And it's just asking, beyond pathology, are there places that you'd like to see synoptic data used in medicine, and why?
Yeah, well, yeah, absolutely. I think across many specialties, subspecialties, but if I look at radiology and reading radiology reports, some can be very succinct and get to the point, and some can be paragraphs long. So a synoptic or structured tool for radiology data would be exceedingly useful. Say, just what are the key points or diagnostic elements I need to know and be able to use when I'm looking at a biopsy or a resection on that patient.
Right, and we know that mTuitive has already done a lot of work with the surgical suite, which is another place that is very amenable to capturing this data. When I teach the residents about how the mTuitive tool works, and I do use it as an educational tool, it's really an inference engine. It's a learning engine that you can literally use to capture any kind of data that you want. So this could be used in a clinic for physical exam structuring, so forth and so on. So I think the real question becomes, where is the value of the structured data, and how and when does that value exceed just kind of running off at the mouth with a soap note or a clinic note? And to me, that's really the question. So obviously radiology, obviously the operative suite and pathology, but you could look at this even in finance. So I'd say the sky is really the limit again.
And I would just add on to Dr. Tuttle and Dr. Eisen was saying that one of the areas that could also help us is specimen sources, and trying to really refine those lists so that it's standard across the board in any structured fashion for anybody who's a physician reporting out, that we're seeing the same lists and we're pulling the same specimen sources from them.
Yeah.
That would be very helpful.
Want to jump in. It's beyond pathology. You can also implement it with the hospitalists in the hospital and pharmacy. The number of antibiotics the patient is taking, correlation with microbiology results, and do they fit the treatment? And it could be concurrent report, which if there is discrepancy, it's flagged. And this probably will prevent of C. diff infection and other acquired problems because of too many antibiotics, so incompatible treatment. But also, as we mentioned, we can understand better our population where we live by knowing what is going with molecular data and the cancer staging as well. Thank you.
Thank you very much. Well, we are getting to the end of the hour here. I'm going to take the remainder of the questions offline. I'm going to, I have a log of those. I'm going to share those with our panelists. We will review them. As always, you can visit cap.org to learn more about electronic cancer protocols, and mtuitive.com to learn about all of their products and solutions. We thank you for your participation today. Look forward to seeing you at future web seminars. Thank you to all of our panelists, and a very good day from the CAP and mTuitive.